Hello everyone. Today we are going to speak about biological databases. To begin with, let me ask you a question. In your opinion, how are biological knowledge and data are generated and be stored? As we know, the most important thesis for applied bioinformatics is the collection of sequence data and its associated biological information from protein function, for example, and protein structure. All these data should be stored in a way that make it accessible and could be used appropriately. Depending on the kind of data included, different categories of biological databases can be distinguished. As we can see, we can find primary biological databases, secondary databases, molecular structure, and genotype-phenotype databases. In this video, we will discuss each category of these biodatabases. For primary databases like UniProt and GenBank. These kind of databases contain primary sequence information, for example, sequence of nucleotide or protein sequence, and accompanying annotation information. For nucleotide sequence databases, the most well-known databases is GenBank, which is perhaps the best non-nucleotide sequence databases available at NCBI. While for protein sequence databases, we can find UniProt. UniProt, which is a consortium built by EBI, the Swiss Institution of Bioinformatics, and the Georgetown University. This consortium aims of developing a central catalog for protein information. Secondary databases. Secondary databases summarize the results from analysis of primary protein sequence databases. For example, here we can find examples of secondary databases: ProCyte, PRINT, FAM, and Interpro. These analyses help us classify of unknown sequence or annotations. Moreover, literature databases are classified as secondary databases. For example, PubMed is a well-known also secondary databases. For genotype-phenotype databases, we can know the relationship, or we can demonstrate the relationship between genes and the biological properties of organisms. For example, of genotype-phenotype databases, OMIA, and phenomic databases. OMIOM stands for the Online Mendelian Inheritance in Man, while phenomic databases is multi-organism genotype-phenotype database which contain data from humans and other important organisms. The last kind of biological databases we will discuss in this video is molecular structure databases, and we can now find protein database, SCOOP, CAT, and PubChem. These databases are crucial for determining crystal structures of biological macromolecules and their functions, for example, enzymes, proteins, and other proteins or macromolecules. Later we will discuss about each of proteins bases and other kind of biological databases which we'll use in applied bioinformatics. How are they significant and how we can use it widely. Now I will ask you a question. Have you ever used one of these databases in your, for example, research? Or please let me down below in the comments. Thank you so much.